Welcome back to online lectures. We are discussing topics enzymology of replication, types of the replication, proof of the replication. In the last sessions, we discussed about replication. What is replication? What is unidirectional replication and bidirectional mode of the replication? In this session, we are going to discuss about rolling circle mode of DNA replication. See, what is rolling circle mode of DNA replication? You see, in the last sessions we discussed that replication of linear DNA molecules on uni and bidirectional mode of replications. In this session, the rolling circle mode of DNA replication can be seen in the prokaryotes, in bacteria, while the DNA replicates through rolling circle mode. The rotation of the replication around the DNA, thereby known as rolling circle mode of DNA replication. Here we can take an example of Escherichia coli, in which plasmid known as F factor can be transferred to F minus cells through the mechanism of rolling circle mode of replication. So here the double-stranded circular DNA undergoes replication and a single copy of DNA molecule transferred to Escherichia coli bacterium. In that cells, now the single DNA molecule undergoes replication and forms double-stranded DNA. So now we will see how this replication occurs. The general machinery, we know that the enzymes required as the same: DNA polymerases, helicases, single strand binding proteins and topoisomerases. So the synthesis mechanism also same. The direction of the synthesis is 5' to 3' direction. Here, during the rolling circle mode of the replication, the 3' hydroxyl group is readily available to enzyme DNA polymerase 3. So, without having any primer, it can replicate and synthesizes new copy of the DNA on the circular template DNA molecule. What are the stages involved in the rolling circle mode of DNA replication? They are of initiation, elongation and termination. Coming to initiation, that means how this covalently closed double-stranded DNA involves in the initiation of the DNA replication, to which the DNA first, the supercoils must be removed with the help of topoisomerases. What is the function of the topoisomerase? The function of topoisomerase is to release the strain in the covalently closed circular DNA to remove the stress and forms the negative supercoils. So, in such a way, the strain is removed and it helps the helicase enzyme to move along these closed DNA molecule to separate the two DNA strands. Here, it requires ATP to break down the covalently closed DNA molecule. So here, the one more protein known as RET-A protein, which is involved in the formation of nick on the surface of the covalently closed DNA molecule. Coming to introduction of rolling circle DNA replication. Rolling circle replication describes a process of unidirectional nucleic acid replication that can rapidly synthesize multiple copies of circular molecules of DNA or RNA, such as plasmids, the genome of bacteriophages, and the circular RNA genome of viroids. Some eukaryotic viruses also replicate their DNA via the rolling circle mechanism. Usually, under the name rolling circle amplification, the mechanism is also widely used in the laboratory in molecular biology research and in nanotechnology. Coming to initiation, rolling circle mode initiates by the production of nick on one of the two strands, producing free 3' hydroxyl group and 5' ends by the action of helicase, topoisomerase and single-stranded binding proteins. So here the nick is formed by A protein and followed by the separation of the strands with the help of helicase, and topoisomerases unwinds the supercoils and facilitates the helicase engine to move along the phosphodiester bonds, and single strand binding proteins which binds to the separated strands and prevents the re-annealing of the strands. In such a way, 3' hydroxyl group is readily available to the DNA polymerase enzyme to initiate the synthesis of the DNA, while the synthesis is initiated by using the inside circle of the DNA as template, and the surrounded circle, 5' phosphate circle, is slowly displacing from this circle. For elongation, the RNA polymerase 3 binds to the 3' hydroxyl group of broken strand using the unbroken strand as a template. The polymerase will start to move in a circle for elongation, due to which it is named as rolling circle model. As the elongation proceeds, the 5' end will be displaced and will grow out like a weaving thread. Here you can see in this picture 3' hydroxyl group and 5' phosphate. While the synthesis is initiated by the DNA polymerase 3, because readily 3' hydroxyl group is available in this replication. So without any help of the RNA primer, DNA polymerase starts synthesis on the unbroken template strand, and the single strand is slowly displacing from this double helical circular DNA molecule as a thread. Coming to termination, at the point of termination, the linear DNA molecule is cleaved from the circle, resulting in a double-stranded circular DNA molecule and a single-stranded linear DNA molecule. The linear single-stranded molecule is circularized by the action of ligase and then replication to double-stranded circular plasmid molecule. Here you can see the picture. A nick is formed, DNA synthesis initiates, and the outer circle of the DNA displaces as a thread, and this becomes circularized, and the nick is sealed by the ligase enzyme, produces a single strand covalently closed circular DNA on which double-stranded DNA is synthesized, on which another DNA molecule is synthesized and becomes double-stranded circular DNA molecule. We can see rolling circle mode of replication in Escherichia coli bacterium, where the two bacterium, one is positive for F factor, another one is negative for F factor. Positive one is known as donor and negative one is known as F minus cell. When a conjugation tube is formed during the mating of these bacteria, a nick is formed on the F factor, allows the formation of 3' hydroxyl group and 5' phosphate group. And the 5' phosphate displacing strand through the cytoplasmic tube reaches the F minus cell, and within the F plus cell, the 3' hydroxyl group allows the synthesis of the DNA on the template strand. And the displacing strand gradually reaches the F minus cell and circularized. Now the circularized single-stranded DNA molecule in the F minus cell undergoes replication and becomes the double-stranded circular DNA molecule. So thereby, in such a way, F factor transfers from donor to recipient through rolling circle mode of replication. Here, F minus cells becomes F plus cells by this mechanism of the replication.
In this image, the DNA from a bacterium has been gently released from the cell. Most of the DNA is from the bacterial chromosome, but small circular DNA molecules called plasmids also exist. For many plasmids, replication is not tied to chromosomal replication. Many plasmids replicate autonomously by a method called rolling circle replication. In rolling circle replication, a replication initiator protein called Rep-A binds to a section of the double-stranded DNA called the origin of replication, or ORI. Rep-A is encoded by a plasmid gene. Rep-A nicks one strand of the DNA and holds on to the 5' end of the strand. The 3' end, with its free hydroxyl group, serves as a primer for a host DNA polymerase to begin to replicate the intact complementary strand. The Rep-A initiator protein recruits a helicase that unwinds the DNA. As the DNA unwinds, it becomes coated by single-strand DNA-binding proteins. As replication proceeds, the nicked strand, which continues to be covered with single-strand DNA-binding proteins, progressively peels off until replication of the intact strand is complete. The two ends of the nicked single strand are rejoined by the Rep-A protein and released. DNA ligase seals the nick in the double-stranded molecule. The single-stranded DNA can now be replicated. A region of the DNA becomes looped, allowing RNA polymerase access to the DNA to form a primer. Host DNA polymerases use the primer as a starting point for the synthesis of DNA. Finally, DNA ligase seals the remaining nick, resulting in a double-stranded plasmid. Each of these plasmids can undergo replication again by the same method.